Good morning, everybody. I'm Pat Swazi. I'm president of the Federation of Vermont Lakes and Ponds. I want to welcome you all to our first winter seminar on emerging aquatic invasive species in Vermont inland lakes. I just want to thank a few people at the outset. I'd like to thank Jack Widnes, chair of our AIS Water Quality Committee, and that committee for helping to organize this. Jackie Sprague, vice president of the Federation and also chair of the events committee, those two committees organized it. A special shout out to Dave Johnson, who's been collecting presentations and keeping everybody organized. And a very special thank you to Meg Modley from Lake Champlain Basin Program, who was instrumental in helping to get this set up today. Just a few words, as so many of you know, and if you haven't heard, I'm sure you'll hear soon. We're facing a problem with aquatic invasive species here in Vermont, with the spread of those that already exist and the lack of funding to prevent that spread and to control infestations that already exist. And we're also facing new threats coming in. And today we're going to focus on those new threats, but keeping in mind that we need to all work together to try to get the state to increase funding and prevent any further spread of any invasive species. So without further ado, I won't go on about that because you're going to hear plenty about that today. I'm going to turn things over to Jack, who is going to do a little bit of housekeeping and also then introduce and get things started. So thank you, Jack. Unmute yourself. No, I got to ask you to do
Sorry about that. Hello, everyone, and thanks for joining us. Thanks, Pat. Well, today's a really special day that we've been looking forward to. We have over 110 people signed up today, which is amazing. This is a hot topic. And we picked these hot topics with the help of you all. We've gotten these ideas in the question and answer period at the end about what topics you'd like to hear about. And our ad hoc AIS committee group for FOVLAP seeks out these topics. And with 110 of you signing up, I think we've picked a good one today, and we have a terrific group of speakers that are lined up. So I want to thank you all for that. In order to continue that, I just want to note that later today in the chat box, there will be a questionnaire placed there in terms of your evaluation of today. It only takes a couple minutes, and we'd be grateful to you if you would fill that out. So I would like to thank the three experts who will be joining us today, talking about these hot topics of potential emerging invasives that are coming in. And also, if you're looking ahead, our next one of these is going to be on the Lakewise program, and it's going to be on February 21st. And we have Allison Marchioni, who's the new director of that, who will be speaking. That'll be a one-woman show, but we'll be taking your questions and comments, and I hope that you'll join us then. Just a couple housekeeping things, as Pat was noting. If you're not speaking, please leave your device, computer on the muted mode. For questions today, we would encourage you to put these in the chat box, but if you prefer to raise your hand and use the hand device in the reaction box at the bottom, that's fine too, and we can have you talk about that. One thing with the speakers is we really need to keep on time, and as you can see from the schedule, we've allotted a fair amount of time for question and answer as well as the presentation. So Glenn Schwartz will be helping me with that, and his wife will be looking in the chat box and helping with that. So unless there's other questions or other comments, I think we're ready to go without further ado. And we'll begin with our first speaker. We could please, who is Kim Jensen. I think a lot of you know Kim. Kim is an environmental scientist with the Lakes and Ponds team. And she is the leader of the AIS Greeter program. A lot of you know Kim through your interactions with the Greeter program, and we welcome her and thank her for joining us. So Kim, please go ahead.
Great, thanks for the introduction, Jack, and thanks for inviting me to speak. I'm going to turn off my video because it's gray in Elmore and I might have low bandwidth. So to do the presentation, which is very large, I'm going to turn off my video. Okay, so I think folks should be able to see my presentation. I'm sure you'll let me know if it isn't showing. We're good?
It's good.
Thanks.
Go ahead, Kim.
So this is the overall aquatic invasive species map in Vermont. We actually finally updated it yesterday. Thank you, Olin, who's our new AIS/ANC permit specialist, and here in the audience. So thank you, Olin. It's nice to have help to do these kinds of things. So unfortunately, it doesn't show the overlaps because some species are found in one water body. So there's multiple species in one water body. But this gives you a general idea of where they are. So the biggest one, the Eurasian water milfoil in red, you can see it's pretty spread throughout the state, minus the center where there's really a lack of water bodies and mountains. Water chestnut, or I'm sorry, curly leaf pondweed is our next contender. And you can see the pictures here. So here's water chestnut. Hopefully you folks are familiar with water chestnut. It's a rosette that's floating and then has a submersed stem as well. And curly leaf pondweed. Sorry, I'm not feeling well today, so I'm a little mixed up. So here's Eurasian water milfoil. You all should be familiar with all of them, but here's Eurasian water milfoil. That is the one that's got more than 14 slight leaves, subleaves along the whorls, and it's found in the 103 now water bodies. And then curly leaf pondweed is the second, 37. Water chestnut, Lake Champlain, plus 34 water bodies. Brittle naiad over here, 11. European frogbit, and have room for the photo, and starry stonewort, and so on and so forth. I'm not going to read all my slides. I think you can see them. We also take care of tracking aquatic invasive animals too. So we have our top contenders there: zebra mussel, spiny and fishhook waterflea, alewife, Asian clam, and rusty crayfish. So when we find a invasive species in a water body, we put them on the list. And this is the Vermont infested water bodies list. Hopefully everybody is familiar with actually everything I'm saying today, so I'm not going to repeat that. You should be familiar with this. So to get on the list, we have to have confirmed with a plant survey that you are on the list, and with specimens and so on. So this list was developed, I believe, in 2011, and Ann from Compile let me know. And on this list, there's also private water bodies. There's multiple regions in Champlain Lake. So over time, the numbers have gone up and down. So this is the water bodies compilation since 2011, when we started tracking these in a spreadsheet. And so you can see wherever there's a little flag was where there was a new infestation. And in some cases, there might have been also a reduction. So that's good news, isn't it? So here's the list, and you can see You know, since 2018, it's been pretty busy. Lots going on since 2018. And you can also see the past year that we have had new infestations found. So since I've been in the program, these events, these early detection rapid response events, were far and few between, it seemed. But for the past 3 years especially, they've increased dramatically. Last year I had 15 calls from folks of a new introduction. Out of those 15 events, and when there is a new introduction, basically my techs and I, since I didn't have a full-time person at this time, we drop everything and we go and we survey. We do usually an eDNA test. We also will do a physical survey and see what the introduction is about. Out of those 15 events that happened this past summer, we have 3 new Eurasian water milfoil, 1 in Eden that was reported by our VIP, and 1 in Half Moon, and 1 in Silver Lake reported by Ann Bove. Thank you all for helping. We also have 3 new water chestnut, and those aren't really recognized in this chart because, well, first of all, it's not in this chart. Okay, oh, here it is, sorry. So there you go, 34, but we actually have 3 new introductions, and those are new introductions in Lake Champlain. So it doesn't really bump up the number except for the 1 site at Arrowhead Lake. That was a new introduction. And then we have 1 that's not on this list, which is Nelumbo lutea, a new species altogether. So this is a really great graph that Chris developed from that data. It visually represents that compilation. So you can see Eurasian water milfoil is our biggest threat. And of those threats, I wanted to show you basically how these numbers relate to how we manage. So Eurasian water milfoil, the status is increasing, as you saw from the former map. And then curly leaf pondweed, it likely is increasing, although it's probably underreported. Water chestnut, big time expanding, probably due to flyways with geese, but that's not a scientifically proven method or vector. European froglet, another one that I'm seeing more and more of, and we don't have any means to control that particular species, even though it's an annual and can be easily harvested. Brittle naiad, also increasing. And then we have some decreases. So yay, good stories. Variable leaf water milfoil really doesn't prefer our water chemistry. It prefers acidic lakes, which is more found in New Hampshire. So we don't have as big as a problem. And then of course, starry stonewort found in 2 water bodies, but for the past few years, we found those plants decreasing. And I'm not sure why. We're still checking as to why that is. And then we have the animal species. Most likely they're increasing, but we don't really have enough staff or enough resources to survey. We do survey for zebra mussel each year at 30+ lakes, and most likely they are in the Vermont side of Memphremagog. I did receive the reports from the scientists who were helping me survey those, and I have yet to compare the 2 maps, but likely they are right on the border. Asian clam also is expanding throughout the Poultney-Meadowy River, and it is in Bomoseen. This is a pretty cool map. I'm going to try to go through it pretty quickly, but it shows you how quickly a infestation can spread. And everybody's thing is interrupting. Okay. So this is Eurasian water milfoil spreading through the state, and the region. And you can see it definitely grows in clusters or expands in clusters and along waterways, the Connecticut River, and also in regions too. So you can see in the Northeast Kingdom, it really took off. So, that's what we're trying to stop with breeder programs, with VIP programs, and with other folks just, reporting things to us. So here's the new species that was found in the Brattleboro region by some naturalists, and they alerted me. My staff went down there and checked it out, and I did as well. And certainly there is an infestation. We're not sure what we're going to identify this species as, whether it's native or not native. So we have a lot of work to do over the winter time to figure that out, because some states list it as native and some do not. So I know this presentation is about the number of AIS infestations in inland water bodies, and I'm not really sure how many inland water bodies are infested or how many species we have. But we do know that Lake Champlain has 51. And so whether inland water bodies have all those 51, I'm really not sure. Again, we don't have enough resources to figure that out. Lake Champlain Basin Program creates this wonderful guide, and we just updated it this past year. So this should be available for folks this summer to provide to the public.
Kim, if you can kind of speed it up, time's just about up.
Yep, I think I have one more slide.
Okay.
So just to talk about animals briefly, the animals, especially fish, they are overseen by the Fish and Wildlife, but we don't know where all the fish are. Again, lack of resources. And then we, through the Aquatic Nuisance Control Statute, we take care or oversee mollusks and snails and crayfish. But again, we started the Vermont Patrollers for Animals program this year to get a good idea of where these animals are with citizen scientists' help. But even that is not enough. So we just want to also look out for other species of concern, and those are some species listed here in different states, and our two next speakers will be talking about these. So I'm going to stop sharing and see if there's any questions. Or maybe I'll keep my screen up. So what are the questions?
So thanks very much, Kim. That was terrific. As you put your questions in the chat, or if you'd rather raise your hand and speak and unmute yourself, we can do that.
Okay, I have a couple questions in the chat. Could the apparent increases be due to the education we have all been receiving? So we now know what to look for, and also the increased boat inspections. Could that be adding to the increased reporting? Yep.
That's a great question. I don't know who asked that, but a great question. There's a timer going on in the background. If somebody can turn that off, that would be helpful. So likely it's just new introductions. I think, yes, that the education has been paramount and knowing where they are, but these are lakes that people have been to, and these are reported by anglers and greeters and VIPs. So I think it's really due to COVID and the fact that there's more people coming here and not aware of the regulations.
Thanks,
Okay.
Kim. Go ahead.
No, I think the other question was about access to slides. That's been answered. And what lakes are the rusty crawfish in? But I think that was also on your chart, right?
Yes, it is in our chart. Yeah. Most of those reports are from the White River, because there's an excellent White River Watershed Association that does a rusty crayfish program that we provide funding for.
So Kim, this is Jack. I have a question. So lake, similar to that? And can you eradicate this? And have there been cases that you are confident that eradication of Eurasian water milfoil is possible?
So to the first question, it really depends on how expanded the introduction was. The introduction in Eden this year, we thought it was a small spot that could be quickly dashed out, diver operated, suction harvested out, or hand pulled actually out. And my staff and I, we went there 3 times, I think, to hand pull. And then they did hire a dash diver to go in because we were finding more and more as we did a survey. So it really depends on the amount of the infestation. We do hope that as we find it, it can find a new introduction, it can be eradicated. But the Silver Lake infestation that Anne found, that was well beyond our capacity to do anything about that. So that is one that's just listed, and there's no management activities as yet being planned in my office, at least. I believe the Forest Service, which land is on, is talking to some partners down there. So that may be happening. And then, I'm sorry, what's your second question, Jack?
I was asking about, eradication. How are there examples of that in the state, and is that really feasible? And
yeah, well, Shadow and Seymour, great examples of people just jumping in and being on it. Chrissy can talk about that, or Beth Torpy. So it can happen. It's really imperative that folks in the community get behind it quickly. And that's what happened at Eden too this year. Like, people were out there volunteering, helping out. And, approximately, I would approximate maybe $15,000 for the first few years. But again, that really depends on the size. And yes, eradication can happen. We're helping Bristol Pond with the eradication of brittle naiad. With water chestnut, we definitely have seen eradication happening with new introductions. So yes, it definitely can happen.
thanks very much. So one thing I just want to remind people is the slides that Kim and the others have been using will be available on the video recording down the road. So if you'd like to have access to those, we will have that. I think there's one other question, Cheryl, in the chat
one more
box.
question for Kim. In your opinion, is there a reason for the slow growth in water fleas infestations?
Yeah, I think our next speaker is going to talk about this, who's more of an expert than I, but I'm pretty sure it's the water chemistry that doesn't support them, and also the cold, that keeps them at bay. So there has been increases and decreases of the population over time, and we see that with other animal species as well. So I think that is the reason.
Thank you, Kim.
Okay. Are there any other questions? If you'd like to either put them in the chat box or raise your hand, we'd be glad to take them right now.
We're a couple minutes ahead of schedule, so if there's any questions.
Okay, well, let's move ahead. Our next speaker is going to be Mike Greer, and Mike is with the U.S. Army Corps of Engineers, and we're going to be talking about hydrilla. Hydrilla is a really feared aquatic invasive that I understand is, and Mike will tell us about this, is approaching us perhaps from the south. A few years ago, maybe 2 years ago, I live on Lake Raponda, and there was an iNaturalist report of hydrilla in Lake Raponda, and I flipped out. As you can imagine, I contacted Kim immediately. She responded, and in fact, it wasn't hydrilla, thank goodness. But this is one that causes fear in everyone. So, Mike, we're delighted to have you, and go ahead with your presentation.
Yeah, thank you. I'm happy to be here. Let me go ahead and see if I can get this shared up. I'm not a proficient Zoom user. So you'll have to just bear with me for a minute. Oh, let's see.
Dave, do you need to help Mike a little?
It may be easier if you can just share my presentation for me. I've got a bunch of stuff open.
Okay, I will. I'll share.
Sorry about that.
No problem.
Okay.
Okay, perfect. Thank you. I'm Mike Greer. I work for the Army Corps of Engineers Research Development Center. I manage 2 of our aquatic invasive species research programs. One essentially is for plants, and the other is for other animals, let's say. Prior to working for the research lab, I worked in Buffalo District, and one of my responsibilities at Buffalo District was managing our hydrilla response in the Great Lakes. And that goes back as far as 2013. So I'm going to just provide an overview of my experience of hydrilla and what is sort of at the doorstep of Vermont. And really not going to try to scare you, but there is plenty of reason to be concerned. And, you know, there's some proactive steps that can be taken to prevent its introduction, and certainly being prepared for a response should you need to respond will make that process a lot more efficient, well received, and hopefully more effective. Next slide. All right, so you know, prior to 2013, I'd really heard very little about hydrilla. It was a plant, a problem that, you know, we really associated more closely with the Southeast and Southern United States. But right around 2013, it became an emerging threat within the Great Lakes in the Northeast. It is on the federal noxious weed list. It's illegal to transport, trade, buy, sell, barter, whatever. Unfortunately, it does get moved around pretty easily through a number of different vectors. It's got huge impacts to our waterways. You know, how we use them for flood control, navigation, hydropower, and then of course, the environmental impacts can be profound, and all of the economic impacts associated with having healthy, usable waterways for tourism, recreation, things of that nature. We did a risk assessment on the Great Lakes, and I'm just going to share that number with you because it's one that I have, and it's relatively credible. But we estimated the impact of hydrilla spreading and becoming established in the Great Lakes to be somewhere between $70 and $500 million annually. So, you know, we kind of use that to help gauge, you know, what's an appropriate response to that kind of problem, right? If it's a small problem, small impact, we might temper our response a little bit more. But within the Great Lakes, and I think that generally applies to other water bodies, the potential for economic impact is very, very significant. Next slide, please. All right, so just gonna walk you through a little bit of hydrilla 101, little walk through the biology. So as Jack mentioned, there is a native lookalike. I don't know what you saw that you thought was hydrilla, but it was likely Elodea. And right here, on the left side, I'm just showing quote unquote typical hydrilla, because we've got a few different varieties in the United States that have a little bit different features, but there's some that are very common. So the plant can grow quite rapidly. The leaves are in whorls of 5 to 10. It varies. The whorls are the collection of leaves around the stem. So there's a close-up of that, and you can also see them spaced out along the stem. One of the defining characteristics of the leaves, besides the number of whorls that we find around the stem, are that it has a spined midrib on the leaf, and it's also got serrated edges on the leaf. It can be a little bit difficult to tell. You almost always have to look at it with like a hand lens or something like that to really see some of these defining features, and sort of compare and contrast that to Elodea. Identifying the plant can also be tricky depending on what growth stage it's in, you know, whether it's early in the season or late in the season. More mature plants, these features will become a little bit more prominent. Younger plants, they're much more difficult to pick out, and the growing tips can sometimes be a little misleading. Its main means of overwintering, survival from one year to the next, is either a tuber that's produced within the sediment. Plants can produce many tubers. Individual plants can produce over 100 tubers. It's a very prolific spreader. Another way that it reproduces is by producing a turion, which is basically a vegetative structure at the tip of the plant that is really not much different than a tuber. It's just less dense, and those turions can break off of the tips, float around, sink to the bottom, and then form a new population. Typically, we don't see too many flowers in the Northeast. That's pretty common with the dioecious hydrilla. That's more in the southern area, but they do or can produce flowers. The other way that hydrilla spreads is through fragmentation, and I'll touch more on that later. But the plant's very brittle, and fragments can create new populations. The native lookalike is Elodea. It's very easily confused with Elodea, again, depending on the growth stage and the person doing the ID. And I've done this many times, and frequently have to ask a coworker for confirmation on whether or not it's hydrilla or Elodea. Next slide. Okay, so as I mentioned, we've got a few different types of hydrilla within the United States. Monoecious hydrilla is what we see more commonly in the Northeast and Mid-Atlantic states. It's native to Eastern China and Korea. That would be all of the green hut codes that you see there, the green polygons. Dioecious hydrilla, we're not sure exactly where it's native to, but it appears like India, Pakistan, China, that part of Asia. And that's very commonly found in the Southeast and Southern United States. It's more closely associated with warmer climates. And then it's really hard to see on this map. Most folks probably can't see it, but we do have a new genotype that was found in the Connecticut River. So far, the testing that we've done, it has only been found in the Connecticut River. And we've done some pretty extensive genetic analysis on plants throughout the Northeast and across the country. And it's just really in the Connecticut River from about the Massachusetts state line from Six Flags down to the mouth of the river. You know, at some point, the saltwater concentration becomes too much for the plant to survive. So we don't see it all the way to the ocean, but it goes down pretty extensively, again, from about the Connecticut-Massachusetts state line all the way through Connecticut. And then, you know, we do see some areas where we get monoecious and dioecious hydrilla. Next slide. All right. Just to give you a little frame of reference, on the left is a picture of the genotype that's found in the Connecticut River. And that photo and mount was prepared by the Connecticut Agricultural Station. It's got some distinguishing features. It's not super easy to tell the difference if you were just to go out and do field observations, but when you put them side by side, occasionally, you know, to the naked eye, you could tell the difference between this unique genotype that's in the Connecticut River and, say, monoecious hydrilla that's more common in the Northeast. One of the biggest defining features is the Connecticut River hydrilla almost always has whorls of 10, usually many more than 5. And you can see a couple of the whorls mounted in the lower left-hand corner of that picture. It's also a pretty robust plant, tends to be really dense at the tips. It's typically a little bit darker green. You know, those aren't really tried and true identification methods, but it is kind of some unique features that make it a little different. Over on the right, you see what's, you know, kind of representative of monoecious hydrilla. It's a lot lighter, tends to be less dense. It doesn't look as robust, even though it is still a very, very invasive plant and can survive in just about any kind of condition. They're very hardy plants. Next slide. All right, so why are people worried about hydrilla? What's the big deal, right? Well, it spreads really, really fast. It is a hardy plant. It can grow under a variety of conditions that our native plants can't. It starts growing relatively early in the season and can quickly shade out some of our native plants, basically outcompete them. You know, it's not too much different than a big family dinner. The first to the table gets the most. And hydrilla is pretty good at getting those resources within the environment before a lot of the native species outcompetes them. And I kind of already mentioned the idea that hydrilla spreads through fragmentation. It's somewhat of a fragile plant. It's brittle. So when boats go through patches of hydrilla, whether it's a motorboat or a paddle boat, that plant's easily disturbed, breaks apart, and then all of those new fragments can settle to the bottom, form roots, and start a new population. That's probably a very, very common method of spread. One of my coworkers is doing some research down at the University of Florida, and they're doing some tests to see how long hydrilla fragments are viable after being on a boat trailer. You know, so say for example, pressed between a boat and the bunk on the trailer, desiccated, and then they put them back in water to see if they can root and be viable. And they can go quite a long time being desiccated, rehydrated, and you know, producing viable populations. So it's very prone to spread for a number of different reasons. Next slide. So I'm going to show a few slides just to sort of demonstrate how easily it's spread and how quickly a small problem, a small infestation can become a big problem, and why it's important to be prepared to respond should you have to. So this graph is specific to Wake County in North Carolina, just one county, not the whole state. They had their first introduction in the early 80s, and 10 years later, that county had a very significant problem, right? And we've seen this pattern repeated over and over and over and over with both monoecious and dioecious hydrilla. It doesn't seem like a problem at first, and then all of a sudden, one day you wake up and it's everywhere. Next slide. So these two maps really tell a similar story. I realize you can't probably read all of the text, but I'll explain it. Pymatooning Reservoir is a relatively shallow man-made lake on the Ohio-Pennsylvania state line. We were assisting them with surveys and management at that lake. We first went out there in 2015, conducted detailed point intercept surveys on the lake. And, we basically ID all the plants, do some statistics. The bottom line is hydrilla was only found in about 11% of our survey locations. The colors on the map show where and what density. So green would be a low density, yellow would be a medium density, red is high density. Okay. Move ahead 1 year. With no management, hydrilla frequency increased threefold, 3 times, right? And that problem continued, right? Until we started management on the reservoir. So you see, not only did the frequency increase substantially, but also the number of points where there's high density hydrilla infestations became much, much higher. Okay, so you really need to look at frequency and density together to start to understand the magnitude of the problem. And it grew quickly, very quickly. Next slide. Okay, so here's an example a little bit closer to Vermont. I don't have the same statistics and detailed information on the Connecticut River, but again, this story is repeating itself. It was first found in 2016. There wasn't a response. I don't think people really understood what was about to happen, but detailed surveys conducted by the Connecticut Agricultural Experiment Station a few years later showed over 700 acres of hydrilla were present in the river. So all of that topped out vegetation that you see on the surface of the water is exclusively hydrilla. And I don't know what the exact acres are, but the hydrilla is ubiquitous from the Massachusetts state line all the way down to where the salt water prevents its growth. It's just mind-boggling that this variety of hydrilla got into so many tributaries, backwaters, and so on that quick. In the picture in the lower right, you see what looks like a little navigation channel. All of the material you see on the left and right of that channel is hydrilla. And really the only thing that's keeping it growing from in that very middle section is the water gets a little deep, the water is a little turbid, a little dark, so there's not a lot of light penetration, so the hydrilla doesn't grow there. But everywhere where there's suitable light penetration, hydrilla is growing. It's a big issue. Next slide. So
Mike,
this saying,
Mike, you got about 2 minutes. You stay on time here, okay?
okay, yep. So the old adage, an ounce of prevention is worth a pound of cure. Absolutely true. It's something that we need to embrace and work towards in all of our waterways. Next slide. In terms of management methods, it's really very limited. And I know a lot of folks won't be happy to hear this, but really the primary means of managing hydrilla is through EPA registered herbicides. Grass carp come with a whole host of problems. Mechanical harvesting tends to exacerbate the problem because it creates more fragments. Environmental extremes can give temporary relief from the problem, but they don't really solve it. We have not identified successful biocontrols for hydrilla yet in the United States, and the only other option is to do nothing. Next slide. And next slide, sorry, there was some animation there I didn't catch. All right, so this slide's sort of specific to New York State, but just for frame of reference, there's over 225 listed herbicides for use in terrestrial settings. And aquatics, there's a couple of dozen, not even. And I've listed all of those products here for your reference, and you can look into those on your own, but not all of these products even have activity on hydrilla. So when it comes to managing hydrilla, we've got a fairly limited toolbox, and we're trying to get creative on how we use those tools and expand the number of tools in the toolbox. Next slide. So should you have to, develop a response, I think these are some really important considerations, right? Your goals, your objectives, how are you going to monitor that and marry that with your treatment? I don't need to go through that in detail, but it's there for everyone's reference, and it follows the template that most lake managers would recommend. Next slide. And I've just listed some additional resources here. Folks that have done a lot of management, public outreach and education, things of that nature. So you may find some of those resources helpful. And I think that's my last slide. Sorry, I had to rush through those last couple.
So Mike, thanks very much. That was terrific. This is a scary invasive that's coming in. And yes, you were right. The story I told beforehand, it was Elodea canadensis, the native waterweed that was seen in Rapanda. And so I just want to give a shout out again to Kim and her team. They really helped us quickly. And this is important for you to know in terms of what Mike just said, is identifying this and then instituting the sort of measures that maybe he'll talk a little bit more in the Q&A here are really important. And Kim and her team jumped on this situation. It was very helpful. The Vermont Invasive Patrollers that the DEC runs is a really important program for identifying these, having volunteers on your lake. So thanks really for a terrific presentation. Do we have any questions in the chat box you can help us with?
Yes, there are a couple questions. I think most of them have been answered, but let's go through them. Jody White had heard that there is hydrilla in Cayuga, and I think Kim confirmed that that is so, and added a link about information about that infestation. Julie Crocker asked if you could define the difference between the monoecious and the dioecious. And that was answered further down, that the mono has two separate plants. I'm sorry, the mono has both sexes in the same plant, and the dioecious has the separate male and female plants. Origin of the infestations. Richard Williams asked if that could be mentioned. Was it through the aquarium trade? And I believe Kim answered that yes, initially it was through the aquarium trade and later through boat traffic. Again, there's a fact sheet with information. Ellen Marsden, she answered the question about the types of plants. How was California able to eradicate its infestations, shown on your first map? Was that through herbicide? That's from Bunny and Ross Westcott. And Meg Modley Mike, can you share a hydrilla management success story like the Erie Canal or Croton River Reservoir? How was that achieved? And
Yeah,
then,
there are success stories. It can be done. It really requires a very rapid response to keep the problem manageable. And we've tried a few different techniques. Early on, it's easy. It's easy. The first couple of years, it's easy. We manage it. We use approved herbicides, giving consideration to the use restrictions and the type of water body we're using it in. Hydrilla's life history leaves it vulnerable to well-timed herbicide applications. And we've kind of figured out all of that science. And, we can very quickly reduce the population by 90% or more within a couple of years of management. What we're finding is because of the tuber bank with monoecious hydrilla, that tuber bank can persist for many years. We're finding that eradicating that last 5% of hydrilla is very, very challenging. And if you're not persistent and very thoughtful about your treatment regime, that can start to get away from you and you go backwards. That's kind of it in a nutshell. That's almost a whole nother presentation in and of itself.
okay. And herbicides were used in California?
Yes, they were.
Okay. And that's also the control choice in the South, yes?
Yes, yep. Yeah, and they did it through very, very aggressive treatment. Folks think about California being sort of risk adverse when it comes to herbicide use, but they understood the nature of the problem and they had a very aggressive multi-year treatment.
Mike, you mentioned the study about the persistence of even dried plants, and you said it was quite long. So what are you talking about when you said its survival was quite long? Is it a day, couple days? Is it a month? Is it the next season? Because this is really important.
No, they haven't published their data yet. We were talking about it when I was there for a visit. It was on the order of a couple of weeks.
Okay. But it wasn't like from one season to the next.
No, not that long.
Yeah.
But certainly, if you pull your trailer out and you've got hydrilla on it and you park it in your driveway for a few days and then go to another water body and it falls off your trailer in that water body, it's reasonable to think that those plants would be viable.
And with Connecticut not too far away from us, we really need to be on our toes. And so inspection of these boats, drying and cleaning is so essential.
Yes.
Okay. Another couple questions. How long are the tubers viable?
They can last in the sediment for 5 years or longer.
Wow. And do the ecosystems recover from successful herbicide treatments?
Yes. Yeah. There's a way to use herbicides that make them a little more selective. They're not broad spectrum, right? They don't necessarily kill every plant in the system, but it does take time for those systems to recover from that type of management.
So, Mike, are you involved in what's happening with the Connecticut River? And can you give us any specifics in terms of, things that are being done there that you'd like to call to our attention to monitor its spread and try to, reduce it?
Yeah, I'm very involved in that. We are starting a demonstration program on the Connecticut River.
Okay.
Basically to, it's a different type. We don't know if it's going to behave the same as other types, and monoecious and dioecious hydrilla don't respond exactly the same way to herbicides, and they've got different life histories, different growth patterns. So we're trying to understand that about the hydrilla in the Connecticut River. Does it respond the same way as other types of hydrilla? What is its life history, so we can identify those vulnerable points in its growth cycle to our advantage in management. So it's going to be a multi-year, fairly robust field demonstration program that includes related research on its life history, phenology, if you will. And as part of that, we will look at some sort of multi-state response team where we can share best practices, research needs, things of that nature. That's very much a work in progress. We just got the appropriation for that work this year in FY23. And that's a $6 million appropriation specific to the Connecticut River hydrilla. So we will be able to get a lot of important work done with those funds.
Can
One
you
more,
say, go ahead, just
sorry.
one follow up. Can you say a little bit of how quickly it's progressing in the Connecticut River? How rapidly is it moving, or does it seem to be static now?
Well, it's everywhere, right? So
And
from
spreading
the
rapidly?
Massachusetts state line down, it's essentially everywhere it can grow. We have not seen it spread further north, and we haven't seen it jump outside of the Connecticut River watershed yet.
Okay. I'm sorry, Cheryl, go ahead.
That's okay. Last question. I have a camp on Maidstone Lake, which is super clean and free of invasive species. We occasionally have seaplanes landing and taking off. Are there any regulations with the FAA and pilots to not spread invasive species? That's from
can't-
Preston Horton.
Yeah, I can't answer that question, but seaplane is definitely would be a concerning vector. I'm going to guess there's no regulations on that though.
So as part of that question, wake boats have ballast tanks. Are they a potential vector? And are you aware of any spread through wake boats where they take in fluid to do their wake sporting and then they dump it in another lake?
It's a big concern, not just with hydrilla, but many other invasive species as well. And again, it depends on the state you're in, but whatever regulations pertain to, boat use and transportation between water bodies, some states have mandatory inspections, others don't. In New York, that's not the case. That's where I live, and I'm most familiar with their boating regulations. But and it's not just boating. It can be rowing, it can be fishermen, it can, there's a whole host of ways that recreation transports invasive species.
Okay, any last questions before we move on?
No, looks like we're clear now.
All right, thanks, Cheryl.
And we're right on schedule.
Okay, Glenn, thank you, and thank the speakers. So our next speaker is Ellen Marsden, and we're going to be switching from plants to animals with the last two presentations. So Ellen is the professor in the Rubenstein School of Environmental and Natural Resources at the University of Vermont. And Ellen, we're so glad to have you here today. So thank you.
Pleasure to be here. Would you prefer that I share my screen or that you try project it from the file that you have?
Why don't you
can
go
share
ahead,
your
Alan?
screen.
Yep.
Gotcha. Give me a moment then to bring that up. No. 1 second. I realize I'm not there. Okay. Are we sharing?
Not yet. Here we go. Here we go. Just a
Oh,
little
there you go.
delay.
Excellent.
Thank you.
And we do that. All right. You should have just one fish image on the screen. So, around gobies, Neogobus melanostomus, basically what I'll do is give you something about the basic biology, how they've spread, where they've spread, what the likelihood is that they're going to get to your lake, and something about control or lack thereof. So this little beastie, it looks pretty large in that photograph. They max out is, and I'll show you in a moment, is on the underside. The problem with gobies is they look extremely like our native sculpins. We have mottled sculpins and slimy sculpins throughout the Northeast, probably not so much in the smaller lakes, but definitely in Champlain and Lake George and the Great Lakes, because they're a cold water fish, and they're very vulnerable to being displaced by round gobies. Can you see my mouse on the screen?
Yes.
Good. I feel like I'm waving my arms in the air and maybe not visible. Gotcha. And so the most distinctive feature of gobies in all of North America, because there are no native freshwater gobies in North America, are these fused fins on the underside of the belly that look a little bit like a disc. And let me give you a close-up of that. Basically, this is the two fused fins. Here's our goby and our sculpin again, now reversed in position. They're preserved, that's why they look terrible. But most fish have two pelvic or ventral fins, depending on what you want to call them. And in the gobies, they've fused together to form something that looks like, but doesn't act quite like, a suction disc. Anybody who tells you they've seen a funny looking fish with a belly that looks like there's a suction disc, panic, right? It's going to be a goby. So they also take different appearances depending on whether they're in spawning condition or not, depending on whether they're male or female. So they live to 4 to 5 years, occasionally 6, but they mature in a couple of years in the case of the females, and longer in terms of in the case of the males. And that is simply because the males do most of the parental care. So they've got more investment in the offspring than the female, and they need more energy to guard the offspring. So basically, not to go too far into the fun biology of this, but they build nests. And when I say build, they really find a cavity underneath a rock or in a beer can or a cinder block or something like that. The male prepares it. Females come in and glue their eggs into the nest, and then the male will spend 5 to 8 weeks defending that nest from all comers until the little ones have hatched. And so that's exhausting to the male, and often they die at the end of that. They need to be big. But they can carry off multiple batches of spawning throughout the summer, which makes them extremely fecund. They can produce lots of offspring in a year, particularly because of this aggressive nest guarding. So this dark male on the right, that is a nest guarding male. They're jet black with a few nice yellow highlights, and very obvious, but you would not instantly confuse them with the normal form unless you'd been pro-warned by me. In habitat, they prefer shallow waters. They're a warm water fish, and they're also in rivers. They like sandy, rocky, open substrate, but with a little bit of shelter nearby. So you rarely find them sort of way out in featureless substrate. There needs to be a few rocks, crevices, something that they can dodge into to hide. They're an extraordinarily aggressive species and extraordinarily hardy species, and they reach very high population densities quickly. Anybody who's snorkeled or scuba dived in areas where gobies are, it's just a little unnerving. It looks like something out of Hitchcock's The Birds. They will all come over and gaze at you, and there'll suddenly be a few hundred gobies around you. Aggressive to other fish species. And so, as I'll mention again in a moment, one of the species that's been most affected by gobies is the sculpins, where sculpins also build a nest or tuck their eggs inside crevices, and gobies are displacing them. They're stealing their nesting sites. They're just bullying them out of the way, and we're losing sculpin populations. In terms of hardiness, and this is very key to the ability of this animal to be spread by humans in something like bait buckets, I'll just give you a scenario. In April, we angled a fish out of the water. The water was probably about 60 degrees Fahrenheit. Saw it was a goby, took the hook off without great care, dropped it into a little pool of water that in the sunlight was probably about 90 degrees centigrade, realized maybe we should take it home, dropped it back into a bucket at 60 degrees degrees. It was fine, which is very unusual for fish. They normally don't take that kind of abuse, either physically or thermally. They are generalist feeders, as most good invaders have a broad dietary ability to eat a broad diet rather than being specialists. So they'll eat insect larvae, benthic invertebrates of various kinds. Very particularly, they will eat zebra mussels. And so in their native zone in the Black and Caspian Seas, they are a native predator of zebra and quagga mussels. And don't get too excited about that, by the way. They won't rid your lakes of zebra mussels and quagga mussels. If you had that many gobies, you'd be worried about the gobies. Fish eggs, fish larvae are a significant portion of their diet when those things are available. And that's one of the other chief concerns about their ecological impacts, is they will eat lake trout eggs, and they'll eat eggs of various species and their larvae. And at high population densities can presumably do a fair amount of damage to any species that's got limited eggs or is in trouble to start with. So this is where they started, the Black and the Caspian Seas in green here. That was their native area. It's not really clear why it took until the 1990s for them to start spreading. But you'll notice that the spread was very rapid in the last 30 years, and it follows rivers. So if there are connected waterways, gobies tend to be homebodies, but then they will take sort of saltatory leaps, and they'll suddenly jump, several kilometers in a week or two, and then they'll stay there for a while. So they tend to stick to home ranges and then do these sudden jumps. And not, to my knowledge, it's not entirely clear why that is the case. In North America, they appeared in 1990, so something triggered this beginning of these invasions, and they showed up in Lake St. Clair. A lot of invasions show up in Lake St. Clair, likely because it's one of the stop places where ships with ballast water coming in from Europe are dumping ballast as they get into the Great Lakes, and it seems to be ending up a lot. Quagga mussels, zebra mussels showed up in Lake St. Clair first. Well, certainly zebra mussels did. From there, they spread extremely rapidly. There was a slight delay while they got themselves started. And then when they started to spread, it took 5 years to get to all of the Great Lakes. And of course, now they're working their way down the river into the Mississippi. Unfortunate, but they're on their way down. The Chicago Canal barrier that was established to stop gobies was too late to stop the gobies. Then they had to find other things that was a good rationale for having that barrier in place. There's lots of things. One of the things I will point out here, actually, I've got it more clearly on another slide. First, let's show the fact that what worries us now, and the reason I'm talking about gobies now, is that they have jumped to Troy. They're in the Champlain Canal, and that means they have a direct conduit. They're in the Hudson River, excuse me, direct conduit via the Champlain Canal to get into Lake Champlain. And of course, from Lake Champlain, we've got all sorts of human vectors that could bring them into smaller lakes in the area and in the northeast. They spread normally downstream by currents. It's easy by drifting. They do work their way upstream actively on their own, and that's why they're where they are. That's why we're worried about the canal. To get to isolated lakes that aren't aquatically connected, probably bait buckets is the major form of transport. This is not something that gets accidentally pulled into a bilge water or accidentally into, say, a bait bucket, the way zebra mussels might. Because they are so hardy and because they tend to steal bait, and that's one of their major nuisance factors, an angler catching a goby by mistake, ripping it off the hook, dropping it in that bait bucket, as I mentioned, it's likely to survive and go, wow, that's a hardy little bait fish I've got in there, put it back on the bait or on the hook, or you take it to the next body of water you're fishing on, it's still alive, might as well use it. So public outreach to say, check your bait. If you've got a robust little fish in there, it's not good. Check its belly, see if it's got double fins in there. As far as we can tell, once they're established, they cannot be removed. Very well spoken about removing hydrilla early in the invasion. Early in the invasion for gobies is probably they're well established because they're not something that just shows up on the surface. They'd only show up if an angler caught them. The only way to kill a fish invasion is something like MS222 or rotenone is better, something that you can overdose them with for the entire system and kill them off. But it's going to kill all fish. And so it's not a selective pesticide or fish poison. You're going to do significant damage with it. There's one case I know of where it was attempted in a tributary to Lake Simcoe. They rotenone the area trying to get a grasp on the invasion quickly, and it didn't work. So troublesome. It might work in a small pond with a lot of effort and willingness to then restock the pond with all the native species or remove some and then put them back in again. This just gives you an idea of some of it, very few so far, inland invasions. These are Finger Lakes, Onondaga Lake, etc. One up here. This was the tributary in Lake Simcoe. It hasn't been so far a really dramatic invasion of inland bodies of, or I should say disconnected bodies of water that are isolated from areas where they already exist. So why are we worried about them? Well, stealing bait is the major nuisance factor. Anglers are constantly complaining in the Great Lakes that they're trying to fish for walleye and all they get are gobies. They do transmit diseases, viral hemorrhagic septicemia, when it was a big deal a few years back in the Great Lakes, they were a vector. Botulism has been linked to round gobies, forming a link between the bottom eating zebra mussels, aggregating botulism spores in their bodies, being eaten by fish and waterfowl, and then transmitting them to loons and other animals that natives that we care more about. That same transmission, that same vector, means that contaminants can go from zebra mussels that tend to aggregate them from the benthos into fish that eat gobies, things like smallmouth and largemouth bass, then into humans. It's not been so far a major human health concern, but it's out there that's being studied. What's interesting along those lines is that many native species are learning that this is a new forage fish. It's good, it's plentiful, they've learned how to catch it, and we've got lake whitefish and walleye and lake trout and a lot of other species that are benefiting now from this big, robust, very abundant food resource. I mentioned the they're outcompeting native sculpins, and they certainly do a number on other benthic fishes like darters, both being aggressive, displacing them, competing with them, and actively consuming them. One of the biggest effects, other than native sculpins, is a problem with bass nests. Bass spawn in the spring, and the males defend the nest, again, like gobies do, from all comers. If a bass male is removed from his nest, even for a few minutes, other fish will rapidly take advantage of that nest by consuming the eggs. In particular, gobies will. So if an angler pulls a bass off a nest, even in a quick catch and releases it a couple of minutes later, it goes back to the nest. In that time, the nest can be completely denuded of eggs by the gobies, and they'll just it's like an explosion of gobies happening. And then, of course, we've got the problem with them doing the same kind of thing with lake trout eggs, because gobies can access the eggs in the habitat in which the eggs are spawned and hatch. So this problem is big enough, by the way, that in 2004, and this is way early in the invasion, the state of Ohio actually closed the smallmouth bass fishery in Lake Erie, even to catch and release, because of the issues of this depredation of smallmouth bass nests. And I'll end before questions. I'll see if this works. Just give you an idea of what I mean by densities and depredation. So you can't see them easily till they move, right? There's a lot of gobies in that shot. They're coming towards the diver. I basically sat there and watched them. They're very curious, right? And they just come in, and they come in, and they move, and they just increase in density. So this only goes on for about half a minute, but you get a sense of how many there are in a unit of area that can increase very rapidly. And with that, and that'll, as I say, finish in a few moments, I'm happy to answer any questions.
So Ellen, thank you
Yes.
very much. I think I'm going to have nightmares tonight after what you've outlined today. I mean it.
Sorry.
I'm a trout fisherman. I associate sculpin as a sign of health of the stream, and now you tell me that its cousins are,
Yeah.
bad actors, and so
Absolutely.
I thought the hydrilla talk was enough, but now nightmares, as they say.
Yeah.
So let's have questions from the. Let's turn the slides off, if you would, please, Dave, and questions from the chat, please.
It looks like the questions have been answered in the chat, but they were, are greeters in Vermont trained to check bait buckets for round gobies? And Kim answered that yes, they are. We get to the answer here. Vermont greeters are trained on the ground goby and ask anglers about bait species in buckets. And then Jody White asked if the assumptions were that if the assumption was that the gobies could not go up the Richelieu because of the current. And Meg answered that river does have more rapids, dams, etc., that may prove more challenging. However, the Champlain Aquatic Invasive Species Task Force, coordinated by the Lake Champlain Basin Program, is working. Did I skip part of that?
I'll just very briefly add, one of the issues we've seen with the Chicago Canal and trying to avoid Asian carp is that boats tend to have a what's called a boundary layer or sometimes an eddy layer near the hull, and a fish can get entrained against the hull and go up with the boat, even though it's on the outside of the boat. So nothing's impossible. It's probably a lower risk coming up the Richelieu, but it's not a zero risk.
And then greeters, lots of kudos for the wonderful presentation. How do we detect gobies? Is it only by finding them, or are there other ways?
I mean, anglers are probably likely to be the first, I mean, unless every local lake association is actively looking for them by putting down traps, etc. It's going to probably be the anglers who see them first, because they are such, bait thieves. They're actually fairly challenging to sample, to capture. They can be caught in minnow traps. They can be caught with electroshocking, which is a scientific gear, only permissible with permits. We found ways to catch them, but you'd have to be using them very intensively to find an incipient population.
All right, well, that kind of answers the question that I was about to read about, sorry, this is jumping on me. Can they be trapped since they are attracted to the diver?
Right, in that situation, I can catch a sculpin any day of the week, but I have never yet been able to catch a goby while diving with the same gear. They're extremely avoidant.
Okay.
Very impressively so.
Meg Modley has offered to do a presentation at another time on the Lake Champlain response to the threat of the round goby. Some anglers say that there are gobies in Champlain, but Jody White does not believe it, thinks they are just mistaking sculpins. Dave Johnson, does the Vermont Fish and Wildlife Division educate the public about goby hazards? And Meg responded that yes, they've received reports on Lake Champlain. Well, that's more about the Lake Champlain. They're using eDNA, trawling, seining, and electrofishing. Okay, so I think that's about it.
Is there anyone who can respond to the question on whether Vermont Fish and Wildlife does education?
Yeah, this is Kim. So they are part of that partner task force that Meg described in the chat. Fish and Wildlife, Vermont DEC, Quebec Ministry, Ellen, a lot of us are all on that task force. So we're all gearing up and getting ready to send out some educational materials and have been working on it for almost a year, it seems.
Yeah, the
Great.
exciting news is that we can report any sightings now through IMAP invasives in both Vermont and New York. So that's a new thing that Vermont has taken on, which is wonderful. We're going to do a very large bait fish campaign this spring with putting signs up at popular fishing areas and launches to have a link for reporting and how to identify round goby, and also to reinforce the message about not transferring live bait.
Okay, and then not sure if Jody's question about whether there are gobies in Champlain. Have we looked for them in Champlain? Has that been answered?
I don't think anybody can say definitively whether they're present or not, but we are looking. U.S. Fish and Wildlife Service was able to do an environmental DNA collection, looking for round goby as well as some other invasives and rare, threatened, and endangered species they're concerned about at 70 different locations, primarily throughout Lake Champlain at the mouths of rivers and popular fishing access sites. The sites also extended into Lake George in New York, down into the Champlain Canal and north into the Richelieu River and the Chambly Canal, which is owned and operated by Parks Canada. So the results of that information won't be known until late February, early March, but we're also doing huge early detection monitoring campaigns in the Richelieu River and the Champlain Canal that are related to rapid response actions for, if found in a certain location, then what action may be taken.
Awesome, Meg. If you have any links that you can add to the chat, that would be great. Thank you for your help.
So Ellen, that was a terrific presentation. We really appreciate that. You know, with the speed with which it seems to be spreading. I don't know if you brought your crystal ball with you today, but I hope so. I just sort of wonder, you know, when you think it may be showing up in Vermont, in Lake Champlain or in other areas. And I just wonder, does this species go into small, cold water, you know, native brook trout areas, so that is going to be a danger at some point, or is that not an area that they generally have as a viable habitat?
Again, their preferences for warm water. At some point, they've just got to tolerate winter like any, temperate zone fish.
Sure.
And they are found in small streams. They are found in rivers. The potential for direct overlap in the summer months, in the fall and spring months with something like brook trout habitat seems a little on the unlikely side. It's not their preferred temperature. So could they work their way up tributaries from Lake? If they got into Lake Champlain, could they work their way up tributaries into those kinds of habitats? I'm just going to make a sort of an educated guess that they're probably going to stop shy of the optimal habitat for brook trout. Okay.
Any predictions on timing, or you didn't bring your crystal ball today?
Well, it's a little fuzzy, I'm afraid. I hate to say it, I'm not tremendously optimistic about Lake Champlain, because there is now that direct connection. And again, they're facing us, as somebody pointed out, from both directions, right? Richelieu or the Champlain Canal. And it's, anyway, I'm not terrifically optimistic. Small bodies of water, both based on the history, you saw that map I showed, and
right,
also the amount of outreach that's already being done and the amount of just knowledge on the part of anglers. We've already got a lot of bait regulations out there. I think your average Vermont angler is more likely to look at a thing in its bucket that it pulled up by mistake and go, I don't think I should do that, because of all the education that's gone out. I think Basin Program and all of the other state programs, et cetera, are doing a really great job. People have their antennas up. So
Well,
that's my hope.
if we could share your presentation with all the anglers, I think they would be hyper in terms of wanting to not have this and do the sorts of things that are needed. Wow.
Absolutely, absolutely.
Yeah.
The one thing that you just have to worry about just a little bit is the anglers off to the edge there. They're going, oh, they're food for bass. Will I get bigger bass? Because we've certainly seen that with things like alewife. That's probably one of the reasons. It may be the reason that we've got alewife now that came into a small lake in Vermont, which is anglers going, oh, we'll get big brown trout if we have alewife. Again, I think we're in a better place in terms of education, but don't make too big a deal about how many fish are eating gobies and getting fatter on them, because that gives the wrong impression.
Yeah, thank you. Are there other questions? Anyone wants to raise their hand? We're certainly, as I said, able to and willing to have you do that. Or are we ready to move on?
And happy to share the presentation. Maybe that's best done through you.
Yeah. Well, we're recording it, so we do
Yes,
have that. Yeah.
All good.
Yeah, Jack, we're still great on time. We're about 4 minutes ahead of schedule. So if anybody has any questions.
Yeah, the presentations will be available shortly on the FOGLAP website.
Yeah.
I can add a little bit of trivia, if you will, on the background of gobies. In the areas where they're native, the Black and the Caspian Seas, there was a commercial fishery for them, and I think there maybe still is a commercial fishery for them. And apparently they're really good canned in a tomato sauce, and they're also dried and salted and served the way we would peanuts in a bar to make people thirsty so they drink more beer. So that's the other side of gobies. Keep that industry where it belongs.
So we've got a minute or two, so I'm going to just have Dave Johnson step in with a little bit of an interlude to talk about what you're going to put in the chat box with the questionnaire of the evaluation before we start with Dr. Stockwell's presentation. Dave?
Yeah, so as was mentioned at the beginning, we do learn a lot at these meetings about what people are interested in hearing in future seminars. So at the end of the meeting, I'll be putting a link in the chat. And before you sign off, if you could click on that link, in about 2 minutes, you can finish a Google Form survey, which will be very helpful for our committee as we move forward and plan these events.
Okay, well, thanks, Dave. All right, so let's move on. So our next topic is going to be spiny and fishhook water fleas. So we saved the best for last. Just kidding. They're all great presentations. So our speaker this time is Dr. Jason Stockwell, and he's the professor and director, again, in the Rubenstein Ecosystem Science Laboratory in the School of Environmental and Natural Resources. And so, Jason, welcome. We're delighted to have you, and we look forward to your presentation.
Well, thank you for having me. I think probably one of the toughest parts of my job is whenever I have to follow Ellen Marsden giving a talk. So just to prepare you. So I'll try to share my screen here. Is it coming up? Okay, great. Okay, so yeah, I'm gonna talk about the spiny water flea and the fishhook water fleas, which have both invaded Lake Champlain in the last decade. First of all, a shout out to Tim Myhook, who is probably a better person to give this presentation than myself, but it worked out this way. So he's provided a lot of information that's part of this presentation. And so both spiny and fishhook water fleas are there. They're both cladocerans, which is a type of zooplankton. If people are familiar with Daphnia, so this is kind of like a water flea, a Daphnia that is much larger and predatory, and you can see that they are they could look like a star in a sci-fi horror film. You know, picture one of these, the size of a building. They're originally from the Ponto-Caspian region over in Europe and Asia. And they are very strong predators of other zooplankton, and to that effect, they can have strong impacts on zooplankton communities. So we're talking about the microscopic animals that kind of link phytoplankton to fish. So they kind of are a critical link between primary producers and then the fish we love to watch and catch and eat. And because of this, their ability to have impacts on zooplankton communities, they can thus have impacts on fish communities. And they reproduce parthenogenically, which essentially means that they're producing clones, kind of like Daphnia or just like Daphnia. And that means they have a high reproductive capacity, so they can reel off a lot of clutches of eggs quickly and exhibit exponential growth. And then typically at the end of the growing season, when the conditions start to get colder, they will produce resting eggs, or also known as Aphippia. And these are basically, they're resistant to environmental conditions. So they overwinter as this, and then when conditions become more favorable the following spring or the following summer, they will emerge from dormancy. And you can see in the bottom figure here, you can see this is an Aphippia egg here. So it's kind of like a seed. So spiny waterfleas first invaded or first discovered in the Great Lakes and Lake Huron in 1984. They quickly made their way to the other Great Lakes and then have worked their way into other inland systems. And this is just kind of a figure that Tim had put together showing their progression towards Lake Champlain over the last 20 to 30 years. We first discovered in Lake George in 2012, and then it showed up in Champlain in 2014. And in between here in the Great Lakes, it showed up in a lot of other lakes. And I'm going to talk a little bit more of the data from Champlain from the last couple of years later in the talk. Ecologically speaking, let's see here. So the impacts of spiny waterflea, which has probably been researched a lot more than the fishhook flea, is that they reduce zooplankton biodiversity in lakes. And here's an example from a small lake in Ontario, north of Toronto, a couple of hours. And you can see that the diversity of cladocerans, again, those species that are like Daphnia, declined sharply when the lake was invaded around 1990. And so you can see a dramatic drop going from 7 to 8 species down to 2 to 3 species of cladocerans in Harp Lake. Now, these type of impacts can induce what we call trophic cascades, where you have a strong, a predator has a strong impact on its prey, and then that affects the trophic level before. So in this case, here's an example of how the abundance of density, the density of spiny waterfleas or Bithotrephes is the genus, has impacted rotifers. Rotifers are microcrustaceans, and they tend to be outcompeted by or preyed upon by other zooplankton. But here we see, with an increasing in the spiny waterflea densities, the rotifer densities go up. And this is because the Bithotrephes are removing the predators or the competitors of rotifers. And so we're seeing a response at a trophic level down. And then this is likely to have other impacts on the phytoplankton community below that. So it's kind of like a series of cascades of positive and negative impacts. And then not only do spiny waterflea and fishhook flea eat zooplankton, but they also have sublethal effects. So they change behavior. And here's a nice example from some experiments done in the Great Lakes region, where on the left axis, there's the depth of the experimental chamber. So it's the surface down to 80 centimeters. And here you can see that in experimental chambers without Bithrotrephes, these Daphnia were predominantly up near the surface during the day. But when Bithrotrephes were present in the chambers, the Daphnia tended to migrate down to the bottom of the chamber. And then at night, you can see that the Daphnia stayed up near the surface like they did during the day without the spiny waterflea, but then they did come up in the presence at night, but they were down during deep. So it's inducing they go down deeper during the day. Increase in their growth rate, in their production. And if you think in terms of like bank accounts and retirement accounts and compound interest, you know, oh, it doesn't look like a sharp drop, but once you start compounding that over generations, it can have a big impact on the overall productivity of the system, which has implications for fish, which like to eat things like Daphnia. So these non-consumptive or sublethal effects are changing the zooplankton behavior, and that these sublethal effects in these experiments in the Great Lakes region could have a much greater impact than just eating the zooplankton. So the change in behavior might have a bigger effect on zooplankton dynamics than just the spiny waterflea eating those directly. The spiny waterflea is also a strong competitor with native invertebrates and fish species. And so in this figure, this is from Lake Huron. What we have here is the production of the zooplankton population in the blank bars on the left side over from May down to October. And then here's the demand or the estimated consumption of different predators on the zooplankton. And what you can see is that the spiny waterflea is in the gray here. And what you can see is that they showed up in Lake Huron, and they're actually, they are a dominant predator. They're consuming a lot more zooplankton than all these other native species, these rainbow smelt, bloaters, and other type of fish, and mysis, which is a large macroinvertebrate, which we have here in Lake Champlain and some deep inland lakes. And so what you can see is that the spiny waterflea is actually consuming a lot of the production of zooplankton, and they're actually outcompeting or consuming a majority of the zooplankton compared to native species. We have a native predatory cladoceran here called Leptodoric kintai, which there's a picture of it there. So that's a native predator, and the spiny waterflea has been replacing that through competition in lake systems in Canada. And so that's another effect on how they're affecting our native biota. And there's some evidence that the presence of spiny waterflea in lakes has a detrimental impact on fish community, the planktivorous, the fish that are eating plankton, which can be your perch, they can be your cisco. There'll be the smaller life stages of things like lake trout and walleye, and you know, every fish is probably planktivorous at some point in its life, just depending on its size. And so here are some examples of some studies done in different lakes around the world where they are finding negative impacts through reduction in growth or reduction in reproductive rates. Lake Maggiore is in Italy, Lake Erie is the Great Lakes, and I've done some work on Lake Superior. There was some work done in Harp Lake, but there was the conclusions were mismatched because of the timing of when they did the study. But anyway, so there can be a negative impact on the fish community. And then there's potential, as you can imagine from what I've been describing, there's changes in food web structure, and then biomagnification. There's some evidence that if you insert a new trophic level into a food web, so we have this new spiny waterflea coming in, it's increasing the length of the food chain. And so when that happens, we get concerned about contaminants that are being biomagnified, meaning that the contaminants get more concentrated each trophic level you go up. And so if we're inserting another trophic level, it's just expanding the opportunities for biomagnification. Okay, so in our region here, the spiny water flea was discovered in the Champlain Canal in 2012. And then it was only 2 years after that when it was first discovered in Lake Champlain in August of 2014. And here's a picture of a spiny water flea with its brood pouch. So it's actually got four young instars of its offspring in that, and it will release it when they're big enough and then exponentially reproduce for the population there. So in Lake Champlain, and this is all data from Tim Hook in the long-term monitoring program, you can see that in early August, there was no Pithecephalus were caught, or no spiny water flea were caught in early 2014, but then it was first detected in the southern part of the main lake here. And then just a few weeks later, it was found all over the main lake, and actually even in Mallett's Bay in the Northeast Arm. So very rapid expansion. And then the rest of that year, 2014, the abundances got really high for a predatory invertebrate. And again, it was all over the lake. So very quick, very rapid expansion upon first detection. Now, the fishhook water flea was first discovered in 2018, and it had a very similar rapid expansion after it was first discovered. You can see here in August, early August, there were no detections. And then in late August, again, it was discovered in a couple different places in relatively low numbers, but a couple weeks later, it was pretty much found everywhere in very high abundances. And again, this is 2018. And then in 2020, it remained abundant. And again, it showed up earlier in the season than we typically expect. You can see there were a couple of detects in early June, but by late June and early July, again, it was in high abundance all over the lake. So impacts on Lake Champlain. This again is mostly work coming out of Tim Hook's lab. When we look at the spiny water flea and the fishhook flea, water flea together, the spiny water flea will typically outcompete the fishhook flea and also prey on them. And so when there's not a lot of food available, we might see the spiny water flea outcompete the fishhook flea. But the spiny water flea has actually been at really low abundances in the last 5 to 7 years since 2016. And the fishhook water flea is actually fairly abundant, fairly steady abundance, is likely due to high predation. So the spiny water flea from fish, the spiny water flea is very conspicuous. And so the bigger you are in aquatic systems, the more conspicuous you are. So if anything can fit you into their mouth, they typically will. And so we think that a high fish predation pressure on spiny water flea is maybe keeping their populations down. There is some evidence that zooplankton populations are shifting their community and their community dynamics in terms of vertical distribution. And this is a master's thesis that was done recently or in the last couple of years. And here's a figure from it. And so this is Daphnia retrocurva, which again is a water flea, very healthy for fish to eat it. But the spiny water flea also likes to eat it. So you can see in 2013, during the day, the Daphnia retrocurva was staying up near the surface where it's warmer, it's growing faster, there's more food up there because of the sunlight. And it remained near the surface day and night. And then in 2014, after the invasion of the spiny water flea, you can see the distribution changed markedly. So they're hanging out in deeper water, colder water, probably less food for them down deep. And then they would come up at night to feed in the surface area under the cover of darkness. This is called a diel vertical migration. So as I showed earlier, the work done in the Great Lakes, this is going to probably have effects on the productivity of Daphnia, which may then have implications for fishes that want to feed on the Daphnia.
Jason, just a couple minutes.
Oh, okay, sorry. I'm about to wrap up here. The fishhook waterflea is still in high abundance since it invaded in 2018. Even though spiny waterflea has declined. And we are seeing, as I showed before, some implications for the zooplankton communities, particularly in late summer when the spiny waterflea and fishhook flea are prevalent. So for just concerning regional lake management, so it would be good to reduce boat traffic from invaded lakes to uninvaded lakes. The spiny waterflea had a progression of about 6 years since it was detected in inland lakes in upper New York State. Unfortunately, the fishhook waterflea, we had no idea it was coming. There was no kind of slow creep. It was in Lake Ontario, and then boom, it showed up here. So boat traffic is probably a way that this gets around. Again, like the round goby, once these invaders get in the system, there's really nothing you can do. And then maybe a way of trying to safeguard is, it's always limited resources, but identifying the most likely routes of the most popular places where people go from Lake Champlain to inland lakes. What are those lakes that are popular? And trying to monitor that boat traffic and maybe focus stewardship and bait bucket checks or boat checks to try to reduce that transmission. So that's all I have. Thank you very much, and I'll be happy to try to answer any questions.
Well, thank you, Jason. That was terrific. So Cheryl, do you want to go to the chat box? Do we have some questions there?
Yes. What methods are used in detection during surveys?
So the Champlain long-term monitoring program is a great example. So using plankton nets and taking samples from lakes, you will typically catch these if they are around, because they are susceptible to nets. Anglers will find them. There was a picture of them on the fishing line. So if they're a high abundance and people are fishing, they're a great detector of both the fishhook flea and the spiny water flea. And then finally, the spines will get stuck in fish stomachs. So if people are fishing and keeping their catch, they can look at the stomach, and if they're eating a lot of them, it will look like a hairball, like a cat hairball. So those are ways you can detect them.
Okay. How well do water fleas survive in live wells and ballasts in the trailered boats?
Well, I can't tell you how well they do. I think they are transferred that way and they can survive. There's treatments are, bleaching and things like that. I think maybe more of a problem is the resting eggs. Is if they get the resting eggs can survive a lot of onslaught of desiccation and cold and stuff like that. So that might be a route of concern.
Okay, and can you please speculate as to why these infestations have been slow in Vermont inland lakes? Do you think it could be pH related?
pH related. Oh, no, because I think any lake that can support Daphnia in general will support these. So, and I'm pretty sure we have Daphnia in a lot of lakes. I don't know why the slowness of it. Maybe there's I don't know what boat traffic is around the state for moving from lake to lake, but perhaps there's not a lot of movement from Lake Champlain, where we do have them, to inland lakes. Other people may have a better feel for that than I do.
That's
Yeah,
my last
Jason,
question.
I'll just say real quickly, the Lake Champlain boat launch stewards do they conduct a risk assessment, as the greeters do with every watercraft they encounter, and exiting watercraft from Lake Champlain headed to uninvaded water bodies are prioritized for detailed inspection and recommended for decontamination for flushing the motor and the bilge. We see a lot of traffic moving back and forth to a number of inland uninvaded water bodies in the state of New York. So that's how they prioritize that. And I think Kim also has, worked on looking at which lakes might be most susceptible to invasion from spiny and fishhook water flea, but along with zebra mussels and many other invasive organisms.
Are there other questions? So one point I'll just bring up that you've sort of touched on, Jason, but I'll just mention that currently the Vermont Agency of Natural Resources is considering a petition to manage wake boats in Vermont. And as you know, wake boats have large ballast tanks. And it doesn't sound like, from what you said, that there's been any actual proof that they can be transferred from lake to lake through ballast tanks, which can't be emptied. There's no filters on these. They retain between about 8 and 25 gallons going from lake to lake. But are there definitive proof of this transport via this mechanism?
Yeah, I think the belief is that, they were first introduced in the Great Lakes, like many things, through the ballast waters of ocean-going vessels coming in. So I didn't mean to imply that they could not survive in bait buckets or in ballast, because I think that is a viable way, and that was probably the mechanism that got them over here in the first place.
Okay, and Meg Modley, in the past, you've talked about flushing out the ballast systems with at least warm water, and has that been proven one way or the other in terms of these particular organisms to eradicate that as a successful mechanism of destroying and eliminating them?
Not specifically, Jack. So the research that's gone on is mostly been focused on Dreissenid mussels. So the zebra and the quagga mussels, which is used kind of as the gold standard for killing early life stages of some of these mollusks in particular. So the approach for all aquatic organisms, because while we're talking about spiny and fishhook right now, we're concerned about quagga and hydrilla and other turions and other things, is for internal compartments to be flushed with 120-degree water for a certain period of time. But that a specific study on the different life stages of spiny and fishhook water flea in ballasted boats in particular has not been conducted to my knowledge.
Okay.
I know that the early life stages or the resting egg stages of the spiny and fishhook can survive desiccation for long periods of time.
Okay, thank you both. Any last questions before we go ahead? Cheryl.
Are they not transmitted on fishing lines?
Yeah, I would think that it'd be hard to transfer them like live individuals, getting them out of water and having them travel. I think the individuals would die. I think the concern there would be if an individual that's on a fishing line has a resting egg in it and then gets thrown into the new lake.
Okay.
Any other questions?
That's it.
Okay.
Right now. Please take the time to provide feedback on the survey that's in the chat box.
Great. And Pat Swazi, do you want to come back, or do you want me to offer a few comments and then you'll close, or how do you want to end this?
Well, I'll go ahead, Jack, and then you can close things up. First, I want to thank everybody. I want to thank Jack and the AIS Water Quality Committee, Glenn and Cheryl, Dave Johnson, Beth Torpy, Martha Winston, Jen Andrews, Jen Hopkins, and the events committee, Jackie Sprague's chair of that, and Dave is there too on that one. Jerry, Jan Parsons on the events committee. I want to thank Meg Modley for her help in organizing this, and then particularly many thanks to our speakers, Kim, Mike Greer, Ellen Marsden, Jason Stockwell. Fabulous event today. I'm really worried. I have a feeling everybody who's listened to this is going away with lots of worries. And I just want to take a minute to ask everyone and remind everyone that the funds for prevention and control of aquatic invasive species are cut for 2023 by $100,000. We have only $350,000 left for the whole state. We need more. There is a bill that was introduced by Representative Carrie Dolan. It's now at the House Environment and Energy Committee, which is the old Natural Resources Committee. They renamed it. And so my ask today is that you take a look at the bill and you take a look at the members of that committee, and you reach out to anyone you can and ask that committee to hold a hearing on the bill. The bill is intended to create an AIS prevention decal program. It does involve fees, and I know that's not to everybody's liking, but it's at least one way we could get into the committee to really make our case that we need more funding. And if you were worried before, this today, I think should make you a lot more worried that we need more greater programs on our inland lakes. We need those that exist to have expanded hours. We need to do a lot. We need to do a lot more prevention work. So I will be sending things out on our email list, but please take a look. We'll be posting information about these. There's several other bills as well that have to do with wake boats that are being introduced this week. So we'll be posting them by the end of the week, but I'll be sending some email out too, asking people to really follow this and reach out to your legislators, because funding is really a problem. And you heard Kim mention several times the lack of resources. The staff in the ANC, the Aquatic Nuisance Control Program that Kim runs, has been cut over the years. Those of us who have worked with it for a number of years remember when there were more staff, and that's another problem. So I will stop there. I want to again thank everybody for coming to this, and thank all of you. So many of you I know have volunteered many hours to prevent these invasions. And I want to thank you for all the work you're doing and will be doing, as I know everybody will be working this summer. So thank you. And I will turn it back to Jack.
Yeah, thanks. I would just really echo what Pat has to say. And we're delighted to have this audience. That is this, we really need in Vermont more money, more effort going into this. And you know what? You're the people that can make that difference. And the reason we have meetings like this, these educational conferences, is to provide you with the information that you need and hope that you will write, because we really need this to happen, to have an impact. And just another plug for the questionnaire. We get the information of what to have at these meetings from you. You really matter. You really do matter. So help us out with that and come up with some good topics for the future. Again, our next one is going to be on the Lakewise program by the new director, Allison Marciani. I think it's going to be terrific. It's going to be on February 21st, and I hope as many of you as possible will join us that day. So thanks to everyone for a great meeting. And I know I learned a lot. I may have nightmares, but it's worth it. So thanks everyone, and enjoy the day. Bye now.